Hello everyone, this is the lecture on architectural patterns of animals, chapter 9 of the zoology textbook. Let's start with a video to get our brain juices going. All right, in this video you will see the process of a very important scientific discovery about how animal bodies are developed from the genetic instructions early in embryonic development. So take note. And while the video plays, I'm going to disappear.
What do we learn by looking at 600 million years of animal history? Evolution's tinkering with mammalness to make whales. In the same way, it's tinkering with fishiness to make tetrapods. And it's tinkering with animalness to make all the different body plans that we see. All these different creatures are variations of the same theme, restated over and over again. The question was, what was evolution tinkering with? One of the remarkable discoveries of the last 20 years is that evolution's not tinkering with the bodies. It's tinkering with the recipe, the machinery that builds bodies. What is that recipe? What is that machinery? It's the genes.
Fossils record the changes in animals' bodies over time. But just how bodies change was unknown. The search for the genetic mechanism of evolution took most of this century. When scientists finally found it, they were astonished by just how simple it was. One of the key players was Mike Levine.
I was, I guess, kind of a weird kid. I always liked bugs. We had a nice big backyard, and I could go back there. It was kind of a sanctuary, and I played with bugs, dissected them, manipulated them. That's really the most pleasant memory I have.
Levine's affinity for bugs led to his study of biology. One insect in particular became an object of fascination.
They have a quick generation time, and they have lots of pattern. I mean, you wouldn't know it if you look at a distance, but when you look under a microscope at an adult fruit fly, you'd be astounded by the number of bristles, the intricacies of their wings, the patterns of their eyes. But the embryos are something else. I do love the embryos.
Scientists had long suspected that embryos held clues to how animals evolve. All embryos start out as clusters of nearly identical cells. But soon, an embryo partitions itself into specialized segments, which develop into the final form of the animal. What controlled this process? How did the embryos know what shape to take? One of the first people to study these questions was a 19th century naturalist named William Bateson. Bateson wrote that animal skeletons revealed an underlying structure of repeating segments. He also observed that animals occasionally developed with some segments in the wrong places.
Insects with legs in the wrong place, crabs where a claw was transformed into a leg, pythons with extra ribs or frogs with extra cervical vertebrae and all these sorts of things.
To Bateson, these developmental errors meant that the underlying blueprint for the animal was being disrupted. He had no idea how it happened, but he suspected that these random changes might provide the fuel for evolution. By the 1940s, scientists working with fruit flies had learned how to cause disruptions in the developmental blueprint by dousing growing embryos with radiation and poison.
And so when they did that, they found flies with changed wing structures, changed legs, and these very special flies that have one part of the body in the wrong place, or a copy of a normal part of the body in another place.
The scientists had triggered the changes by damaging the fly's DNA. Within each cell of the developing embryo is a chain-like molecule called DNA. The experiments showed that DNA was somehow causing the embryo to divide into segments. But how? Scientists were just beginning to grasp that the DNA itself was made up of segments, called genes. The question was, how did the genes shape the body? One researcher, Dr. Ed Lewis of Caltech, studied this question for 30 years by crossbreeding thousands of flies. Lewis's work led him to a controversial idea. He proposed that a surprisingly simple mechanism was shaping embryos. He wrote that each segment of the fly was being directed to grow by a single gene. A small set of genes, a kind of genetic toolkit, appeared to be laying out the entire body.
And as you looked at these genes, you said, this one affects this part of the body, this affects the next part of the body, and this affects the next part of the body. That was an astonishing observation.
It was astonishing because it seemed too simple. Nobody else thought single genes were powerful enough to control something as complex as the structure of the body. Skeptics argued that Lewis's idea was guesswork. Of course, he had never seen the genes, because the techniques to do so didn't exist.
From the 1920s to the 1970s, it was not possible to physically isolate any specific gene. That opportunity first became available, fortunately for me, at the time that I was a student. And so many of us thought, wow, we can finally dig in there and identify these really mysterious genes.
Levine enlisted his friend and fellow scientist Bill McGinnis. The first gene they went after had an unusual name, Antennapedia, which means antenna leg. The gene was thought to control the growth of legs. When the gene misfired, flies grew legs in the wrong place, on their heads in place of antennae. In normal flies, legs grow from the midsection, the area called the thorax. So Levine and McGinnis decided to hunt for the gene in the thorax of a normal embryo.
The expectation is that Antennapedia would be active, expressed in the thorax, the developing thorax of the embryo. But who knew?
Levine and McGinnis had to do something no one had ever done before. They had to find a way to see a gene in action.
We wanted to light up the gene. And it was very painstaking work.
The project called for new and untested methods.
At first it didn't work very well, and there were a number of technical problems to solve.
The team had to find a delicate balance of radioactive probes and toxic enzymes. Too much of either would destroy the embryos.
The process was not very gratifying on a day-by-day basis. Unbelievably tedious.
It took months of trial and error.
People often said, you know, you should try something else. You know, this is too long shot. You know, you're just wasting your time. But we kept going.
Finally, late one night, all the work paid off.
And there was this moment when we saw that the gene was turned on in a band in the middle of a very early embryo. This had never been seen before.
The Antennapedia gene was acting like a master switch, turning on the segment of the embryo that would become the thorax. The implications were mind-boggling. If single genes like Antennapedia could define whole segments of an animal, these genes were acting like architects of the body. And if one of these genes turned on in the wrong place, striking changes to the body could result. It seemed that Lavina McGinnis had uncovered the genes responsible for the evolution of bodies. But there were still doubts. The work had all been done in fruit flies. What about other animals? Did they use the same mechanism to build their bodies? An answer would come from Switzerland. In 1994, Walter Gehring of the University of Basel isolated the gene that triggered the growth of eyes in fruit flies. The gene was called eyeless because flies without it developed with no eyes. Gehring knew of a gene in mice that worked in the same way. He wondered, were the two genes the same?
And this question we tested by taking the mouse gene and putting it into fruit flies to see whether flies can understand the message of the mouse.
Gehring replaced a fly's gene for eyes with the mouse gene.
And to everybody's surprise, the mouse gene works perfectly well and can induce a compound eye in the fruit fly.
The fruit fly grew normal fruit fly eyes using a gene from a mouse. Not only did the two creatures use the same mechanism, they used the same gene. This was the mechanism behind extra wings, legs sprouting from heads, and Bateson's deformed animals. The century-long search was complete. The genetic engine of the body's evolution turned out to be a tiny handful of powerful genes.
So what this means is in some ways, some sense, evolution is a simpler process than we first thought. When you think about all of the diversity of forms out there, we first believed that this would involve all sorts of novel creations, starting from scratch, again and again and again. We now understand that no, that evolution works with packets of information and uses them in new and different ways and new and different combinations without necessarily having to invent anything fundamentally new, but new combinations.
Suddenly, the commonality of form among animals was understood. Animals resembled each other because they all used the same set of genes to build their bodies. A set of genes inherited from a common ancestor that lived long ago.
And what we see now among all the animals are just variations on a body plan that existed 500 million years ago.
All right, I am back. I hope you were paying attention. That was a monumental discovery in Evo Devo. All right, let's go back to our PowerPoint. So one of the things that came up in this video was that the fly embryo understood the instructions, the mouse instructions for making an eye. This came up in the Evo Devo song in the video that I played for you last week, when the artist was saying that my eye protein can make you see out of your bomb, right? So if you put some eye proteins in the posterior of a fly embryo, it will grow eyes in its butt. So that's amazing, right? Even if those initial eye proteins are from a different animal. All right, let's get going here. So zoologists recognize 34 extant phyla in kingdom Animalia. The animal kingdom is also called Metazoa. And here on top of the slide, you see the 34 animal phyla that are currently recognized. Most of the semester, we're going to spend exploring the different animal phyla, but let's remember that those animal phyla are not just completely disconnected entities. That little graphic there I got from the inset of your textbook right here, as you can see. And remember that all of those 34 animal phyla are connected in this intricate tree of life, a phylogeny that connects us all to our common ancestor. However, the 34 phyla that remain are really the survivors of about 100 phyla that appeared about 600 million years ago during the Cambrian explosion. Cambrian explosion was not a literal explosion, it was really an adaptive radiation in the Precambrian oceans that during which there was rapid evolution and all the animal phyla appeared. The Cambrian explosion was perhaps the most important evolutionary event in geological history. Now, what makes one phylum different from another phylum is that each phylum represents a distinct body plan. It's important to note that no new animal phyla have appeared since the Cambrian period. So all of that differentiation, that evolutionary differentiation of animal body plans occurred more than 540 million years ago, which was the onset of the Cambrian. Okay, now, animals are organized. Their bodies are organized. And we recognize 5 grades of organization, each more complex than the one preceding it and building on other grades in a hierarchical fashion. So the first grade of organization is protoplasm, right? Just that the components within a cell are organized so that the cell itself is an organized unit. Some organisms reach only that grade of organization, and those are the unicellular protozoans. If we're talking about eukaryotes. Some other organisms have a more complex grade of organization where different cells get together and work together to provide life to the organism. At the most basic level, in kingdom Animalia, it is the sponges that reach that grade of organization and no higher. So sponges are in phylum Porifera. They are animals that only reach the cell or the cellular grade of organization. The next one is tissue. In this grade of organization, different cells organize and groups of cells engage in a common function. Some animal groups only reach this third level of organization And an example of that would be the jellyfishes in phylum Cnidaria, where they have true tissues, but not true organs, because organs is the next level of organization, in which different tissues come together to form structures that we call organs to engage in a common function. Some animals only reach that level of organization. For example, the flatworms in phylum Platyhelminthes. They'll have a brain, they have other organs like eyes and a gut, but no true organ systems, because organ systems is the fifth and last level or grade of organization. And most animal phyla have that level of complexity where they have organ systems. For example, a skeletal system or a muscular system or a nervous system. In fact, the last third of this semester, we're going to spend exploring the various organ systems. Now, the next thing that we have to discuss when we're talking about animal architecture is symmetry. Some animals, as you've seen in lab, have no symmetry, like this bat sponge. In fact, this bat sponge belongs to phylum Porifera, and poriferans or sponges lack symmetry. Some animals have radial symmetry. Basically, they can be cut in halves, but in any number of axes. An example of an animal with radial symmetry is this coral polyp or sea anemone or jellyfish. Those are animals that belong to phylum Cnidaria. There's another group of animals called the ctenophores or phylum Ctenophora, like this comb jelly, that also have a special type of radial symmetry called biradial symmetry. And together, phylum Cnidaria and phylum Ctenophora make up supergroup Radiata. Lastly, there are some animals, most animals actually, have bilateral symmetry, meaning that they have two mirrored halves if you were to cut the animal along the anterior posterior axis. All animal phyla that have bilateral symmetry belong to a monophyletic supergroup called Bilateria. I'll explain what monophyletic means later in the next lecture, but basically all animals with bilateral symmetry come from or descend from a common ancestor. Now, interestingly, animals with bilateral symmetry have a head, and this is called cephalization. So bilateral symmetry is always associated with cephalization. And this is advantageous because with radial animals, they can interact with their environment in all directions. But with bilateral animals that have a head, and in their head is a brain that can sense the world, that is advantageous for forward movement. All right, the next topic that we're discussing in this lecture of animal architecture is body cavities. Remember the gastrula from last lecture? So if we were to take a longitudinal section through the gastrula and open it so we would see inside, it would look something like this. We will see this single-celled layer on the outside called the ectoderm, the single-celled layer on the inside called the endoderm. Remember, the blastocele is that fluid-filled cavity in between, or that is inside the blastula. So here it is, even in the gastrula. And then soon after the gastrula forms, there will be the origin of mesoderm, right? This middle germ layer. Now, what happens to that germ, that middle germ layer, is very important. There can be three things that happen. One path is that the mesoderm can completely fill the blastocele. That results in a body plan that we call the acoelomate plan. Or a second path is that the mesoderm can partly line the blastocele, and so there's still an inner body cavity that is called the pseudocoelom. The third path that the gastrula can take is that the mesoderm can completely line this new body cavity called the coelom. That is a true body cavity, right? So the coelom is the body cavity, which is why animals that lack a coelom are called acoelomates, and animals that have what looks like a body cavity, but that cavity is not completely lined with mesoderm, that cavity is called the pseudocoelom, or like a fake coelom. Now, don't worry about these two, schistocoelus and enterocoelus. They're basically two different paths that get to the same destination. What I want you to remember are that some animals are acoelomates, some are pseudocoelomates, and some are true coelomates. And these diagrams illustrate what happens early in embryonic development. But whatever path an animal takes is going to have profound consequences for the rest of its life. And it is going to influence what the animal will look like even as an adult. So here we have some cross sections of adult animals. The textbook example of a acoelomate animal is a flatworm, phylum Platyhelminthes. So if we take a cross section of a flatworm, we will see this outer layer, which is derived from the ectoderm. We'll see that there's a gut lined by this cell layer, which is derived from the endoderm. And in between, the whole body is packed by what is called parenchyma, which is a spongy mass of space-filling cells, all derived from mesoderm. The textbook example of a pseudocoelomate is this nematode roundworm, phylum Nematoda. If we take a cross-section of this roundworm, we will see that the outer skin is derived from the ectoderm. The lining of the gut is derived from the endoderm, just like all examples here. However, we will see that this pseudocoel, right, this body cavity is only partially lined with mesoderm, only here on the outside, not actually on the inside. And I want to contrast that with the body plan of a true coelomate, here exemplified by this earthworm as the representative of phylum Annelida. So here again, if we take a cross section of the earthworm and look inside, we will see the ectoderm, or the outer skin, which is derived from ectoderm, the lining of the gut, which is derived from endoderm, and this body cavity, which is completely lined by this tissue called the peritoneum, which is derived from mesoderm. So again, a coelom, a true coelom, is a body cavity in triploblastic animals that is completely lined with mesoderm. This is a an illustration from a different textbook that pretty much does shows the same thing. Okay, so we have the acoelomate body plan. Here's a cross section of a flatworm, phylum Platyhelminthes. You see that in between the ectoderm and the endoderm is this parenchyma, just completely covered by the spongy cell mass derived from mesoderm. If we take a cross section of this roundworm, we'll see that the body cavity, in this case called a pseudocoelom or pseudocoel, is only partially lined by mesoderm. And if we take a cross section of this earthworm, we'll see that the body cavity here called the coelom is completely lined by tissues derived from the mesoderm. Now, earlier in the semester, we discussed that all animals start as a zygote, right? The union of a sperm with an egg. Once the zygote is formed, it will quickly undergo cleavage or cell division and form a blastula. Remember that? All animals form a blastula. However, some animals don't form in any germ layers because they don't go through gastrulation. And in that case, there are no germ layers that are formed. The entire body is just formed out of the blastula, and the only phylum that exemplifies that strategy is phylum Porifera, the sponges. Most other animals go through gastrulation, so they form a gastrula, and once the gastrula is formed, then you have this inner germ layer called the endoderm. Some animals don't develop a third germ layer past that, and then their whole bodies are then formed just from those two germ layers, ectoderm and endoderm. Because there's no mesoderm created, there are just those two layers, we call those animals diploblastic. The examples of that are phylum Cnidaria and phylum Ctenophora. And the rest of animals go through blastulation, they go through gastrulation, but in gastrulation, a third germ layer forms, the mesoderm. So we call all of those animals triploblastic because their bodies are formed from 3 embryonic germ layers. Now remember that in the beginning of gastrulation, that initial hole in which the blastula begins to invaginate, that hole is called a blastopore. And the fate of the blastopore is very important. In many animals, that blastopore becomes the mouth. And when this tube, the forming gut, punches all the way through, forming a second hole, that second hole is the anus. Those animals we call protostomes. Proto meaning first, and stome, like stomata in plants, right? Meaning a hole or a mouth. Meaning the first hole becomes the mouth. Protostomes. Some protostomes are acoelomate. They don't have a coelom. Some protostomes are pseudocoelomate. They have a pseudocoel that is only partially lined by mesoderm. And some protostomes are true coelomates, meaning they have a true coelom that is completely lined by mesoderm. And yet in other animals, the blastopore winds up becoming the anus. And so as this forming gut punches all the way through, it is the second hole that becomes the mouth. We call those animals deuterostomes. Deutero means second, and stome means mouth, so it's the second hole that becomes the mouth, meaning the first hole becomes the anus. chordates, which is the phylum that we belong to, are deuterostomes. So we develop anus first. All right, let's talk about segmentation. Segmentation in zoology is also called metamerism. And this is the serial repetition of similar body segments along the longitudinal axis. In zoology, each segment is called a metamere. Now notice that segmentation permits evolutionary modification of each segment, because each segment can be thought of semi-autonomous from other segments. We'll see that later on when we explore phylum Arthropoda, both in lecture and in lab. And the last thing that in our discussion of animal architecture is that all the tissues of the body can be thought of as coming in 4 flavors. There are 4 kinds of tissues, right? 3 embryonic germ layers will develop into 4 different tissue types. So I want you to study this figure 9.7 in your textbook, so you understand that how different kinds of tissues belong to different types of tissues can be differentiated into these different subtypes. For example, this is easy muscle tissue. There are 3 kinds: smooth muscle, cardiac muscle, and skeletal muscle. All nervous tissue belongs to, all nerves are made of nervous tissue. That's simple. Epithelial tissues are tissues that line stuff. So for example, our skin is made of epithelial tissue. The lining of our gut is made of epithelial tissue. So that's kind of easy to remember. Or maybe a little tricky is connective tissues, which come in different kinds. Let's remember that bone is a living thing, and bone tissue is one kind of connective tissue, as is blood. So again, study figure 9.7 in the corresponding text so you understand these different kinds of tissues and the structures that they form. So here are the sections that you should have read, and if you didn't, you will read. And when you get to 9.3, just focus on this figure 9.7 and the text that corresponds to that. All right, I'm going to stop here. Goodbye.